Lumpy skin disease is an economically important viral disease of cattle, mostly confined to Africa. Outbreaks being reported particularly in southern, eastern and central Africa. It has also occurred very occasionally outside the continent, namely in Madagascar, and in 1989 in Israel, where an outbreak caused severe losses. The disease is of economic importance because it results in a morbidity rate of from 5 to 45% and a mortality rate of usually less than 10%. It causes permanent damage to hides and prolonged debilitation in recovered animals and gives rise to temporary or permanent cessation of milk production as a result of mastitis. Both bulls and cows may become temporarily or permanently infertile, and abortion may occur in about 10% of pregnant animals. Lumpy skin disease is caused by a capripox virus of which the prototype strain Nettling was first isolated in 1944 in South Africa. There is only one immunological type. Mature capripox virions have an oval profile, and their average size is 320 by 260 nanometers. It grows in a wide variety of cell cultures and in chicken embryos, as well as on the chorioallantoic membrane of embryonated hen's eggs. Lumpy skin disease is a systemic disease, and the virus is therefore present not only in skin nodules, but also in lesions in other tissues and organs of the body, and in the saliva and semen. The virus is remarkably stable, and it can persist in necrotic skin nodules for more than a month. All age groups and breeds of cattle are susceptible. The Bos Taurus breeds with thin skins, such as Friesland cattle, appear to be more susceptible to the disease than thick-skinned Zebu-type breeds, and the clinical signs in calves are generally more severe than in mature animals. Clinical disease has not been reported under natural conditions in wildlife. However, giraffe and impala are highly susceptible to experimental infection, and antibodies to the virus have been demonstrated in free-living African buffalo. The reservoir or maintenance host of the virus during inter-epidemic periods remains an enigma. Circumstantial evidence suggests that biting flies such as Stomoxys and Musca species play an important role in the mechanical transmission of the infection. Outbreaks of the disease usually occur in late summer and autumn during years of above average rainfall. Other methods of spread which have been suggested include iatrogenic transmission through blood-contaminated needles and ingestion of saliva-contaminated food. Viral replication in pericytes, endothelial cells, and probably other cells in blood vessel and lymph vessel walls causes vasculitis and lymphangitis in some vessels in affected areas. The vasculitis in severe cases may result in infarction. The clinical signs of typical cases of lumpy skin disease are characteristic. It is important to realize that only 40 to 50% of animals develop generalized skin lesions after infection. Many cases are subclinically infected. Generalized eruption of skin nodules usually appears 7 to 19 days after inoculation of the virus, and viremia persists for 4 days after the initial febrile reaction. Typically, cattle develop a biphasic fever response 2 to 4 weeks after exposure to the virus. They remain febrile for 4 to 14 days, during which time they may develop inappetence, salivation, lacrimation, and a mucoid or mucopurulent nasal discharge. Lacrimation may be followed by conjunctivitis, which may ultimately result in corneal opacity and blindness. In the majority of animals, the superficial lymph nodes are enlarged. Skin nodules appear before or during the second rise in body temperature, 4 to 10 days after the initial fever response. The nodules, which are randomly distributed and range in diameter from 5 to 50 millimeters, involve both the skin and subcutaneous tissues, and sometimes even the underlying musculature. The number of nodules may range from a few to several hundred in severely affected animals, and are usually fairly uniform in size in the same animal. They are well circumscribed, firm, round, and raised, and are particularly conspicuous in short-haired animals. In long-haired cattle, the nodules are often only recognized when the skin is palpated or moistened. In most cases, there are nodules in the skin of the perineum and mucosa of the vulva. Several nodules may fuse to form large, irregularly circumscribed plaques. Skin nodules that do not resolve rapidly either become indurated, in which case they may persist as hard lumps or sitfasts for 12 months or longer, or desquamate as a sequester, which leaves a deep ulcer partly filled with granulation tissue that often suppurates. In most cases with generalized skin lesions, the legs are swollen due to subcutaneous edema, and the skin may become necrotic and slough, leaving large suppurating wounds, which may extend into underlying tissue, such as tendons and tendon sheaths. This results in lameness in the affected leg or legs. A prominent feature in many cases of lumpy skin disease is the appearance on the muzzle and the nasal and buccal mucous membranes of round, initially raised plaques that frequently ulcerate. In bulls, lesions may occur in the skin of the scrotum and mucosa of the prepuce and glans penis, but necrotic lesions may also be found in the testes, resulting in orchitis and testicular atrophy and fibrosis. These animals may become temporarily infertile, or more rarely, permanently sterile. Similarly, lesions in the reproductive tract of cows may result in infertility. Nodules may form in the skin of the udder and teats, and when the parenchyma of the udder is involved, as it frequently is, it may result in mastitis and severe economic losses. If extensive necrosis occurs in the upper respiratory tract, secondarily infected necrotic tissue may be inhaled and result in pneumonia. Stenosis of the trachea may result, following healing of lesions with scar tissue formation some weeks or months after infection. A tentative diagnosis of lumpy skin disease can be made on the characteristic lesions in the skin and visible mucous membranes. The diagnosis can be confirmed within hours by electron microscopic demonstration of virus in negatively stained preparations of biopsies taken from scabs, affected skin, or mucous membranes. Virus isolation from blood collected in heparin during the early febrile reaction and from scabs of the skin lesions is also possible, but it may take 7 to 14 days. The complement fixation and virus neutralization tests can be employed to measure the level of antibodies to the disease in convalescent sera. The clinical signs and lesions in mild cases of lumpy skin disease can easily be confused with pseudo-lumpy skin disease caused by bovine herpes virus 2 infection, which is a much milder disease than lumpy skin disease. The skin lesions are more superficial, and it has a shorter clinical course. Other diseases or conditions which could be confused with lumpy skin disease are those in which skin lesions develop, such as besnoitiosis, insect bites, Demodex infection, and onchocerciosis. Control of lumpy skin disease in Africa is essentially limited to vaccination of cattle. If the disease is introduced into a country free of the disease outside the African continent, quarantine and slaughter of all cattle in the affected area should be considered. 2 vaccines are available for the immunization of cattle against lumpy skin disease, namely, one which contains the attenuated Netlung strain of the lumpy skin disease virus, and the other because there is cross-protection between lumpy skin disease and sheep and goat pox viruses, contains attenuated strains of the latter viruses. The sheep or goat pox vaccine has the disadvantage that it can only be used in countries where sheep pox or goat pox is endemic, as the vaccine could otherwise provide a source of infection for susceptible sheep or goat populations. Both vaccines confer a lifelong immunity following a single inoculation. Approximately 50% of cattle develop a swelling 10 to 20 millimeters in diameter at the point of inoculation, and this may be accompanied by a temporary drop in milk yield in lactating cows. The swelling disappears within a few weeks. Immunity after recovery from natural infection is lifelong in most cattle. Calves younger than 6 months of age, whose mothers were either naturally infected or immunized, possess colostrum immunity and should not be vaccinated to preclude interference from maternal antibody. Calves born to susceptible cows are very susceptible and therefore should be vaccinated as soon as possible in the face of an outbreak. Lumpy skin disease is one of the many African arthropod-borne infectious diseases that plague cattle, but is one that can be controlled effectively by the use of a vaccine.